cool. So thanks very much to Stephen and to the organisers for having us back to talk this year. So Sophia and I are both research scientists based at AstraZeneca, so not too far away, just a building across the road. And we're here to talk today around thermal shift methods, with a particular focus on methods that we employ ourselves for the purpose of high throughput screening. So I thought I'd just start by introducing what HGS is and what we do. So within HGS, we sit within the wider Department of Discovery Sciences, and we comprise a very early stage of the process that goes into discovering a potential new drug. So once targets have been identified and validated as having a particular role in a disease process, we're responsible for the rapid screening of large compound libraries to identify chemical series which are active against those targets. In terms of the compound libraries, we screen AstraZeneca's internal compound collection. We have a collection of 1.6, 1.7 million compounds, and we try and identify things which are active against targets. So in terms of active, we mean kind of they might modulate the activity of that target, or they might just bind to that target. And then these compounds are then taken forwards to later stages of the process to be optimized into potential new drugs. So a good analogy is that HGS functions like a large funnel. So we carry out very quick, rapid screening of large numbers of compounds. And then as we progress through the HGS cascade, these numbers are gradually confirmed and narrowed down until we end up with tens to hundreds of compounds comprising up to 4 chemical series, which are then progressed to later stages. And in order to carry out this very rapid screening in a reliable way, we rely a lot on robotics and automation. So if you come past the ground floor of our lab across the way, you should be able to see these robots, hopefully with green lights on, running our assays for us. So what kinds of assays do we run within HGS? So the kind of assays that we run have really evolved from the kinds of targets that we've been required to screen. So in the past, we've had quite a lot of catalytically active targets with well-known binding sites. So we've been able to develop assays to monitor functional activity, for example, substrate turnover, or designing probes which bind to known binding sites on that target and looking for displacement. However, during more recent years, we've seen a real shift, as these graphs highlight, towards what will be classed from a drug discovery perspective as more challenging targets. So these can be quite hard to prosecute using traditional assay technologies. So they might lack any sort of known structure. They might be catalytically inert, so we can't monitor functional activity in an assay. They might contain disordered domains, or they might lack any sort of known binders or tool compounds, which we'd normally use to validate assays. So this is quite challenging. So in order to tackle these new targets, we've had to evolve our assay technologies, and we rely quite a lot now on biophysical or affinity-based methods, which just identify binders to targets. And then those binders can be used in innovative ways for new modalities, such as, you can develop them into PROTACs and then target maybe a functionally active target for degradation instead. So this is quite cool. Some of the biophysical methods that we employ within HGS are ASMS, which is an affinity-based mass spec assay, DNA encoded library screening, or DEL screening, which uses DNA tags present on each molecule, which are then sequenced to identify a molecule that's bound to your target. And we also employ high throughput thermal shift methods, so DSF and SETSA, which is going to be what we're going to focus on for the rest of the talk. So before we get into things, I thought I'd just give a quick recap on the principle behind thermal shift assays and how they work. So thermal shift assays detect changes in the conformational status of a protein brought about by heating. So this is just a really simple model of the unfolding equilibrium that exists between targets in their native folded state and their unfolded state. And thermal shift assays are able to discriminate between these two states and monitor how that unfolding equilibrium shifts in response to heat or temperature. And this can be really useful in understanding information about ligand binding. So if I just add a ligand binding equilibrium onto this model, for the most part, ligands will bind to your native folded state of the protein to form a protein ligand complex. And in doing so, they are able to stabilize this native folded form and shift the unfolding equilibrium in this direction. So through monitoring this unfolding equilibrium, thermal shift methods can indirectly derive information about ligand binding. And that's kind of, in very simple terms, how they work. So this is a bit of a timeline of different thermal shift methods and when they were introduced. So the principles behind protein unfolding have been studied since like the late 1880s, 1800s. But it wasn't until more recently that the role that heat played in this unfolding was discovered. And in the 1960s, there was the introduction of the first differential scanning calorimeter. I'm not going to focus too much on this because I think it was covered by Stephen in a previous lecture, and I don't know too much about it myself. But essentially, it measures excess heat capacity associated with conformational changes in the protein brought about by heating. So DSC was really great. It allowed measurements of protein unfolding to be undertaken more widely. However, it requires quite high sample volumes, quite a lot of protein, which limited its use for kind of screening purposes and for screening large numbers of compounds. However, more recently, there was the introduction of a fluorescence-based thermal shift assay, which is kind of commonly known as dye-based DSF or just TSA, thermal shift assay. And this was applicable to screening in 3 at 4 well plates with much lower sample volume. So this really opened up the possibility of using thermal shift assays for the purpose of high throughput screening. And it's something that we currently utilise ourselves within HGS AstraZeneca. So what is dye-based DSF and how does it work? So like DSC, it measures the thermal stability of isolated protein targets, but rather than monitoring heat capacity, it measures the fluorescence emission of extrinsic dyes which are present within your sample. And we typically use a hydrophobic dye called Cypra Orange to carry out these measurements. So what happens is, while your protein's happy in its native folded state, dye molecules present in the sample are exposed to the aqueous environment. However, when you gradually increase along the temperature gradient, you start to unfold your protein. Hydrophobic regions within that protein's core are exposed and able to bind to the dye and interact, which increases the quantum yield of the dye, and you see this increase in fluorescence signal up until the point where your protein is completely unfolded, it starts to aggregate and signals quenched again. So this phenomenon typically results in a sigmoidal denaturation profile, which we term a thermal melt curve. And the stage at which half the protein population is unfolded is referred to as the TM, or the melting temperature, and this is something that we monitor throughout the assay. So through monitoring the TM, or the change in TM, the delta TM, this can provide a measurement of the extent of thermal stabilization of the target under different conditions. So some of the advantages of dye-based DSF, as I mentioned, it's convenient, sensitive, and high throughput. You can use it for protein QC, so to check that your protein's happy and properly folded. You can also use it for buffer screening, so assessing which buffers might be more beneficial for protein stability, which can be useful for long-term storage and crystallography. However, as I mentioned, we utilise it to understand information about ligand binding. So ligands will bind to the native folded form of your protein, increase its thermal stability, and this manifests as a positive shift in TM. So that's how we apply it. So this slide, I thought it would just be useful to introduce some of the considerations that you might want to think about if you were developing your own dye-based DSF assay, as we've kind of been through the process. So 2 things you might want to consider are the compounds that you're screening and the plate types that you're using. So within HDS, we screen our regular DSF collection, which is a collection of around 100k compounds. And these are designed, because we're using it for hit binding, they kind of maximise diversity sampled across the whole compound collection. So they have a lot of structures which are representative of different chemotypes within the collection. However, we have limited inclusion of some compounds from that collection. So compounds with poor solubility at 100 micromolar, which is our primary screening concentration. We've seen that these result in a decrease in our assay response. So we've kind of excluded those. Also those which we know might interfere with the assay. So compounds which are intrinsically fluorescent or maybe hydrophobic, they can interact with the dye that we're using. So we've limited inclusion of those as well. We're actually in the process this year of redesigning our DSF collection, because while we've tried to limit compounds which interfere, there are definitely still things in there that we regularly pick up. And there's also stuff in there that we see completely quenches the signal as well. So we're redoing our DSF collection to make it more optimal and to really make use of the compounds that we're screening and save time and resource following stuff up that's kind of artifactual. We can use a variety of different PCR plate types. They're all commercially available, which is really great. You just need to use a specific PCR plate so you can have efficient heat transfer from the thermocycler into your sample. And these are two plate types that we've tested and we've settled on. So the light cycler plates from Roche and the frame style plates from AstraZeneca seem to work well for us. So another important consideration is the type of dye that you're using in the assay. There are many commercially available dyes that can be used for DSF, and this is a really nice paper and table which summarises the different types of dye. As I mentioned, we use separate orange, and this is widely favoured for dye-based applications, specifically for screening, because that has quite a high signal to background in the presence of native folded protein, and it has quite a high emission maxima as well. So it emits at wavelengths between 500 and 610 nanometers. And this is quite useful because if you do have compounds which are intrinsically fluorescent, they tend to fluoresce at lower wavelengths, which minimises interference. A big disadvantage of Cipro though is it's not detergent compatible. So if you have any sort of hydrophobic buffer components or detergent within your sample, this might not be the dye for you, but there are other dyes. So there's this DCVJ molecular rotor dye and this dye Proteus dye, which have been cited in the literature to be able to be used in cases of high detergent. So they might be two to consider. We also tend to focus on screening protein targets using DSF. You can also use it to monitor RNA and even ligand binding to RNA. So two dyes that you can look at in that case is this cybergreen and this ribogreen, a single strand specific RNA stain. And we've actually got a student in the department at the moment who is working on building cascade assays for RNA, and she's looking into dye-based DSF using these two dyes. So not too much to report at the moment, but maybe next year we can come back and share some learnings from that as well. There's also been quite a cool paper which was published this year from Wu et al. And it basically describes a platform that they've built to streamline protein dye pairing for DSF. I'm not sure how to pronounce this, but PADSF. And essentially, yeah, they've described in this paper a number of dyes which could potentially have broader applicability than Cipro Orange, and even potentially produce better assay, better data quality, basically. So there's a few people in the department who have taken a few dyes from this paper and are screening them against a panel of different protein targets to see whether they offer any benefits. So that's something else that we can maybe update on next year if we come back. So yeah, in terms of the protein that we use for the assay, we typically use between 1 to 5 micromolar of protein. So it's quite a high amount of protein. And we'll typically assess this in a titration with dye before we run the assay, just to make sure we've got optimal conditions in terms of dye and protein, and we're getting a decent transition and signal. We use 5 microliters per well, which is quite a low volume, but when you scale that up to the number of compounds that we're screening, that quickly builds, and we typically use between 50 and 150 mgs per screen, which is a lot of protein. And this does actually limit sometimes the applicability of DSF for certain targets, particularly those which are more difficult to express and purify. So those might be more suited to a sector approach, which Sophia will talk about a bit later. And it's the main reason we screen an 100k collection for DSF and not larger numbers of compounds. In terms of reading your plates, you can use any real-time PCR instrument, which is great. We use the Quant Studio 7 Pros. We use a temperature range of 25 to 90 degrees, and most proteins are completely unfolded at 90 degrees. And we use a ramp rate of 1 degree C per minute, which is a fairly typical ramp rate for a sort of DSF experiment. So that was a bit about assay considerations, but how do we actually carry out the analysis? Specifically, how do we calculate this TM value that we monitor in the assay? So as a reminder, the TM is the temperature at which half the population is unfolded. And there are a variety of different ways that you can calculate the TM. Some are more robust than others. The method that we use is calculating the smooth first derivative of the raw melting curve signal or the fluorescence with respect to temperature. And the TM in this case is the inflection point of that curve. We use this method because it's quite robust. It results in quite a reproducible TM value, which is important for determining our assay stats and activity cuts. You don't need to fit a curve, so it's quite a simple method. And crucially, it can be really useful for calculating the TM from multiple different sort of transition shapes. So you don't need this typical sigmoidal melt profile to be able to calculate the TM using the derivative, which can be really useful in cases where you have multiple transitions in your melt curve. So this is an example of 2 targets that we screened a couple of years ago. and there's two transitions in this curve, it could be quite hard to see that from when you just look at raw fluorescence with respect to temperature. However, when you look at it using the derivative, you can clearly see two inflection points in that curve corresponding to two different TM values for the different transitions. And this can sometimes happen if you have a protein with multiple domains that unfold at different rates. They can have two TMs. And it can be really interesting, if you add ligands into the mix and see them preferentially bind and stabilize one TM value, and you know what domains these TMs correspond to, you can use this to derive information about where those ligands might be binding. So it's quite a cool application of DSF if you have that information to hand. In terms of calculating the TM, we use Gene Data's screener software to calculate it for us. You can calculate the derivative of curve in GraphPad Prism, or there's a variety of open source analysis tools that you can also use, and these can be used to fit more complex models to your curve as well. So if you do have a typical sigmoidal curve, you can calculate the Boltzmann, you can fit a Boltzmann equation to that curve to calculate the TM as well. So in terms of actually physically running the assay day to day, it's quite a simple assay to run. This whole process is now fully automated. So essentially we take assay ready plates, which already contain our compounds as singles per well. We dispense our fluorescent dye on top of those compounds using this echo liquid handler, and that uses pulses of acoustic energy to transfer nanoliter droplets into each well. So that's quite a cool method that we rely quite a lot on within HDS. We then add our protein, which has been incubated in buffer. We spin the plate to make sure everything's at the bottom of the well, add a clear plate seal, just to prevent any evaporation at high temperatures, and also obviously to make sure we're not disrupting the fluorescent signal that we generate. Then we incubate for 30 minutes, just to make sure that equilibrium's been established and ligands have had a chance to bind. And then we'll heat and read using the real-time PCR instruments that we've got. So typically it takes us around 2 to 3 weeks to screen 100k compounds at single point. The actives from that are then taken and replated in order for us to carry out a confirmation screen, which is basically just a retest of those compounds to make sure that we see a significant shift again on another occasion, and that we see a dose response, which can just give us confidence that the compounds are binding to our target in a real way. And then we typically validate these binders in orthogonal assays. So we have a dedicated biophys team who will follow up these compounds in assays such as SPR. It's kind of the most common assay, but we also use NMR and LCMS as well. And these are just kind of higher fidelity assays, which gives real confidence that they're real binders. So in terms of our throughput, this says 30 plates a day. We've actually recently pushed it to like 40, 45. So it's about 14k compounds a day. It takes around 14 hours. So I mentioned this is fully automated. We're actually pretty confident in the reliability of this automation now, after running quite a few screens. So we tend to leave it into the night. So while we're like having our dinner, the robots are still hard at work, which seems a bit unfair, but I guess it's what they were made for. So this is just some example data from a target that was screened a couple of years ago. We'll call it kinase X. So it's just an example of some single concentration data from a primary screen. So you can see we've screened just close to 11,000 compounds in this run. The well number is on the x-axis, and then the TM calculated using the derivative method on the y. So you see we've determined an activity cut for this run, and this is what we've determined to be a significant shift based on the variation in neutral control wells, and that's just our protein target in the absence of any ligand. And this, we typically set this, it always seems to land at about a 1 degree TM shift, and that's kind of the consensus in the literature as well as what's deemed a significant shift. However, we have had really tight assays in the past where we've brought that a little bit further and got further into the noise to bring more compounds through. But you can see the majority of compounds here are sitting around that 45 degree mark, and that's the TM of the protein in the absence of ligands. So most compounds are having no effect, which is probably what we'd expect from a hit binding assay at this stage of the cascade. But there are quite a few compounds that sit above that threshold. We could automate this analysis and take all of those through confirmation. However, it can be quite useful to manually interrogate the traces and look at them visually. particularly in cases of compounds which produce a very large shift in TM. So this is an example of one of those compounds. So when you manually look at the trace, in the absence of ligand, it's given this nice, typical profile. With ligand present, you see this really high initial fluorescence, and this can be an example of a compound which might be interacting with the dye, and it kind of looks artifactual. We can't discern a reliable TM measurement from that trace, so we normally tag this as an artifact and exclude it from downstream follow-up, just to save on time and resource in terms of following these up. We do also see quite a few compounds which induce a negative shift in TM. This could be for a number of reasons. So it could be maybe binding to a less stable form of the protein, or preferentially binding to a less stable form. So it could still be having a real effect. However, the problem is we see quite a lot of these compounds in screening runs. Can be non-specific effects as well. So it can make it quite hard to follow these compounds up when there's so many. So we tend to, and also we tend to be more interested in compounds which stabilize the native folded state of our protein. So we tend to deprioritize these negative shifters in favor of compounds which produce positive shifts in TM. And we can always go back and kind of look into them more at later stages if needed. So this is an example of one such compound, a typical active. So you can see that it's producing about a 5 degrees shift in TM, and the trace in the absence of ligands looks very similar to the trace with ligand present. And this is, example of a nice compound which we'd flag as active and take forward to later stages of confirmation. So as I mentioned, at confirmation, we're just looking to confirm significant thermal shift again on another occasion and see this nice concentration dependence when we run a 5-point dose response. It's kind of important to note at this stage that the extent of the TM shift doesn't necessarily correlate to the affinity of the binding for that compound. So ligands with an equal KD can result in very different TM shifts. And in the same way, you can have the same TM shift result from ligands with different KDs. So we tend to use this confirmation run as essentially just confirmation, and then limit sort of any KD or affinity determination to downstream biophysical assays, which carry out that for us. So this is just to mention a little bit about the effects of dye addition. So obviously we're adding a dye into our sample, and when you add anything into a sample, it has the potential to interfere with that system. So I've just added dye binding equilibrium onto the model that we were talking about earlier. So because it's a hydrophobic dye, it binds to preferentially binds to the unfolded form of the protein because it's binding to hydrophobic regions in that core. And in doing so, it kind of shifts the unfolded equilibrium towards this unfolded state. And you can see that in the experimental data that we've generated here. So as you increase the amount of dye added up to 15 times, so for orange dye, you can see an increase in fluorescent signal, which, kind of expected to add in more dye into the sample. But you can see the TM has decreased quite significantly. So there's a shift of -4 degrees. So because of this, we tend to minimize the amount of dye that we include within the assay. And include the minimal amount, which will allow us to get a decent maximum signal for our trace. So we tend to settle on between 2.5 and 5 times dye within the assay. But this is just something to think about, I guess, if you're setting up your own. So more recently, a technique has been introduced which alleviates the need to add an extrinsic dye into your sample, and this is known as label-free or nano-DSF. The throughput is still not quite there with this for us to be able to use it in a screening sense. However, we do use it for protein QC in other departments, and we actually use it to QC our proteins and make sure they're properly folded before we run them through other affinity-based methods such as ASMS and DEL, because having happy pure protein is really important for those methods. So I thought I'd just touch on it briefly. So nano-DSF or label-free DSF monitors the intrinsic fluorescence of tryptophan or tyrosine residues present in your protein target. And most commonly what you see is internal tryptophan residues, which are present within a hydrophobic environment in the protein's core. As the protein's heated, they're exposed to the hydrophilic solvent environment, and this causes a change in the polar surroundings, I guess, and causes a red shift in fluorescence emission or a shift in the emission towards higher wavelengths. So through monitoring the ratio of emission at these two wavelengths, 330 and 350 nanometers, this provides a measurement of the TM, or the melting temperature. So some of the pros of DSF, it's a much more direct approach than dye-based DSF, as you're not adding this dye and this second binding equilibrium into the mix. It can be used for protein QC, as I mentioned, for buffer screening, to assess the relative stability of different protein batches or mutants, and you can also use it to look at ligand binding, but on a smaller scale than we would do for dye-based DSF. A major advantage, I guess, is that it's applicable to a wide range of samples, including those containing detergent. So this has been used to study membrane proteins and stability of that sort of target. In terms of disadvantages, it requires a target molecule to contain tryptophan or tyrosine residues. And depending on the number of residues present in the sequence, it can be a lot less sensitive than dye-based DSF. It's not appropriate for ligands with fluorescence emission in the tryptophan region. And as I mentioned, that the throughput is currently limited. So you do need specialist equipment to monitor these two wavelengths over time and with respect to temperature. The most commonly applied instrument, I think one we use ourselves, is this Prometheus Panther, developed by Nanotemper. And this has a throughput of 48 samples in parallel, which is still quite a high throughput, but for us, yes, it's still not quite there for us to be able to screen 100K compounds quickly. There is actually a new technique that's been introduced by a company called Protein Stable, and they've said that they could run nano DSF in 3/8 ball plates. So I think we said last year that we would look into this. We actually haven't looked into that yet, but if anyone was interested, their website would be a good thing to check out. But that could be quite an exciting new development, which would enable, screening using nano DSF to be more high throughput. So moving a bit more forwards in the timeline, a really exciting new technology has been developed that allows thermal shift measurements to be undertaken within a cellular context, which Sophia is going to talk about in a second. And just briefly before that, more recently, there's been this new technology introduced. It's still very much in the development phase, so Nanotemper are developing it at the moment, and a few of our team are looking into it with them. But it's kind of called optical unfolding, where you have your protein target tagged with fluorophore, and they use a laser to very quickly heat that protein and induce unfolding. And a big advantage or potential advantage of this is that it's a lot more sensitive, and you can potentially use much lower amounts of protein, which might open up the applicability of DSF for much larger numbers of compounds and for those targets which are more difficult to express and purify. So this is quite cool. Again, something that we can maybe share more on when we've generated the data next year. But I'll just pass on to Sophia, I guess, to talk a little bit more about cell-based thermal shift.
Cool, thanks, Poppy. Yeah, so I'm Sophia. So I sit in the cell screening team within HTS. So I'm going to talk about the cell-based thermal shift assays. So cellular thermal shift assay, or CETSA, it was originally invented by Pelago. That's a spin-out company from the Karolinska Institute. It is the same principle as protein-based methods, except that we're within the context of the cell. So you heat your cells containing your target of interest, and your target will unfold. And the idea is that compound binding will decrease the rate of unfolding, so you'll get a shift in your TM. So the principle is the same, but the challenge here is, how do we detect the target protein within the cell? So there's lots of different methods of doing this. The gold standard in the field is to use Western blot, tried and tested. Don't fix it if it ain't broke. But it's obviously very low throughput, so we will use this when we are down to very low compound numbers, and we're looking to characterize those or confirm those. Another option that is used a lot in the field is mass spec. So this is used in the context of looking at the whole proteome, so treating cells with, challenging them with one or a few compounds and looking at the interactome there. So again, that's not suitable for our high throughput screening setup, where we're looking to test many compounds at once. Some of the methods are sort of starting to get more high throughput now, so things like reverse phase protein array, high content imaging, these are plate-based methods. The advantages of, for example, high content imaging is that you can start to interrogate the phenotype of the cells upon compound challenge, as well as just looking at the target of interest. And then in high throughput screening at AZ, we've mainly looked at these two technologies. So the first is alpha screens by Revity. It's antibody based. It requires 2 antibodies targeting your target and beads, conjugated beads, and these act as a donor and acceptor. And essentially, when you excite the donor bead, it will promote the release of singlet oxygen. And if your acceptor bead is in close proximity, this will produce light at a given wavelength. So this is a highly sensitive approach, and it's quite high fidelity in the field. However, it's very expensive, and of course, it requires antibodies. And the whole point of these intractable targets is that they're very novel. There might not be any antibodies reported in the literature for these. So for these targets, we need a different approach. And so what we've been using at AstraZeneca is this split luciferase reporter system via HiBit. So this is a Promega technology called HiBit Scepter, which we've been coining BITSA. It relies on tagging your target of interest with an 11 amino acid HiBit tag using CRISPR or a lenti system. And this HiBit tag binds with very high affinity to what's called large bit. And then this forms the reconstituted nanoluciferase enzyme, which is active. And this can overturn luciferin to generate light. So in a HiBit Scepter assay, we heat the cells, treat with compound, heat the cells, and then we add in the lysis buffer and the detection reagents and read the luminescence. And compounds that are bound will stabilize the protein and increase the amount of luminescence there. So some of the pros and cons of this technology. So compared to the antibody-based methods, it is a relatively simple HGS amenable assay protocol. The antibody-based methods require a lot more addition steps, and because it's bead-based, you get problems with like clogging of the valves and things like that, so you have to be careful with that. We also forego the need for antibodies here, which obviously opens up a lot more possibilities for intractable targets, and you don't have the problems of epiturbance. And because we're not using antibodies, the reagents are relatively inexpensive. No technology is perfect, obviously. So the problem or the major downside here is that you need your engineered HiBit cell line. This can be quite challenging to make, and it can take time, so you have to just factor that in. And also this technology is quite sensitive to or susceptible to artifacts, so you have to have a rigorous downstream deconvolution cascade, otherwise you'll be swimming in artifacts. So we can set these up in lots of different ways. So like the protein-based methods, we can perform melt curves, but because this is an endpoint readout, so we add the lysis buffer and detection reagents, you cannot monitor your target over temperature gradient. So how we have to do these is to perform discrete experiments at each temperature and then build the melt curve. It's quite laborious. It's not what we would call high throughput, so we don't use it for screening. But we do use it further down for sort of confirmation of our compounds further down the cascade. The other thing that you can do is select a single temperature point and then run a compound concentration curve. This will give you an idea of your potency, although like Poppy mentioned, potency and TM shift doesn't necessarily correlate. But yeah, you can use that to, for example, rank your compounds and provide information for the chemists so that they can triage effectively on that. And then because we're in the context of the cell, you can do some fun things if you want to. So you can alter that context via pushing the cells into different activation states if you want to. You can, knock down proteins, knock out proteins. You've got quite a lot of options there. And then I'm not going to talk about lysate-based Scepter, but you can run these assays in lysates, and the benefit there is that you've overcome the challenge of the compound having to get into the cell, and B, you are less prone to artifacts or off-target effects, because when you're in a whole cell, you might have a compound acting upstream of your target, which would then look active, but actually it's off target. So you negate that when you're in lysate format. So in high throughput screening, for our primary screening assay, we select a single temperature to run at. So this would be called an isothermal setzer. The temperature point we have typically gone for has been TM plus a few degrees, so that you have the majority of your protein unfolded. And then when you treat with a compound, a tool compound, you get the maximum signal in your assay, and this becomes your assay window. When we look at that data in terms of our screening data, will be 1 day's worth of screening data, so say 25,000 compounds, what you'll see is that the majority of the compounds, like in the DSF, sit at or below the neutral controls or the DMSO treated controls. And then you have a small number of compounds that are sort of increasing within that assay window, and you can set your active cut at a sensible point to call actives. So this is the theory, but in reality, we are very frequently faced with having no tool compounds for our targets. When you have no tool compound, you don't know what your assay window is, you're blind, and it can make it very, very challenging to set these assays up. Where do you select your temperature point? And so we've had to come up with a way to sort of model this as best we can. So how we do this is we do some simulations in Prism. So we first perform a melt curve on the automation. So we try to collect as many data points as possible. So what we'll do is we'll set 10 plates up with just DMSO in, and then each of those plates will be put through the assay, but at a different temperature, so we can build our melt curve. And then we put this data into Prism, and we can plot a Boltzmann, and then we can apply a right shift to the Boltzmann to the TM, say for a 1 degree, 2 degree, 5 degree stabilization event. And this will allow us to get a prediction of what our assay window might look like at a given temperature. So you can now predict your assay window. What we've also learned from lots of previous experience is that the noise that we see around our data points is directly proportional to the level of signal that these are at. So we can extrapolate the noise from our real melt curve at a given signal level, and we can extrapolate that onto the simulated stabilization events. And what that enables us to do is get a prediction of the assay window, prediction of the noise, and from there we can get our Z prime, which is what we love to see in HTS as a measure of assay robustness. And we like our Z prime to be greater than 0.5 in order for us to take an assay through. So how this data looks like in real life. So we've used this for 2 projects. The first, target A, so we plotted the predicted Z prime across a range of temperatures there. What you can see is that in this assay, between 51 and 53 degrees, we have a robust assay where we can detect even a 1 degree stabilization event. So we use this to set the temperature for screening at 53 or 52 degrees. And we knew that we would be confident to be able to detect those small shifts in the TM. On the right is another example. So here we found that there was one temperature around 43, 47, 48 degrees where we could detect a 5 degree stabilization event just. But other than that, we couldn't detect anything. So we actually use this data to go back to our colleagues in assay development and say, we can't trigger the HTS and risk, wasting a lot of time, money, reagent when we have an assay that we don't have confidence in is robust. So that's a really powerful tool that we've been using to prosecute these challenging targets. So I said earlier that these are simple assays to run. I guess that was in the context of the more complex antibody-based methods. This is actually one of the most challenging assays or complex assays that we run in HTS. It involves an awful lot of kit, multiple plates, and it wouldn't be possible without our fantastic automation team and automation equipment that we've got in the disk. So the process is that we dispense cells into what we call assay ready plates. So these 384 well plates with our compound library arrayed in each well. And then we incubate that for an hour at room temperature. This allows the compound to hopefully get into the cell and bind to the target. We then perform our heat shock step using an on-deck thermocycler by Inheco. And this is only a 2-minute step at the isothermal temperature you've decided for your assay development. We'll then briefly spin that just to remove any condensate that's formed on the lid, and then we'll add the Promega lysis and detection reagents. So that's the lysis buffer mixed with the large bit and the furamazine. And then we do a hard spin, and this spin will pellet any of the aggregated unfolded protein that is going to potentially interfere with our readout. And then we'll transfer the supernatant, 10 microliters of the supernatant, to a fresh plate and read the luminescence in a plate reader. And we carry this out on this platform at the bottom here. So this is called a CoLab platform. So it's essentially a centralized robotic arm that is surrounded by micro docks, which contain all of the bits of equipment above. And it can move the plate through the entire assay protocol when it's working. And it means that we can run about 80 plates a day in an 8-hour day, which is about 25k compounds. Unlike the DSF, we're not at a confidence level where we would be running this out of hours yet, but we would like to get to a point where we can do that. So in terms of how this looks, in terms of a target cascade, so the most recent one that we've done was this target A, this intractable transcription factor. Think big floppy protein, no known binding sites, no catalytic activity. So we used high-bit sensor to screen 500, just over 500,000 compounds against this target. So we did this at 30 micromolar single concentration and at the temperature point we've defined from our modelling. And from this we got 700 actives, very low hit rate. Normally we're looking at 0.5 to 1% hit rate from our typical cell-based screens. However, given the knowledge of the protein, or more the sort of lack of the knowledge of the protein and the challenges around this protein, we actually weren't surprised to have this low hit rate. So we then went into concentration response. So we did 10-point concentration response curves for all 700 compounds, and a lot of them dropped out. So we had 150 actives by this point. So it just shows that single point screening is noisy, and it's just a way of funneling. You have to do it to get those numbers down. But yeah, concentration response as soon as you can. So yeah, we had 150 actives that gave a dose-dependent response in the assay. And then we triaged these in our high throughput melt curve protocol, which again was happening on that pod system. So this got the numbers down by about half again, and then we had 72 actives, which was a really a good number to go into these downstream assays. So SPR, when protein generation was close to impossible, and the biological assays they were running were on quite primary sort of cell lines. So it was good that we were able to pass on quite a small number of compounds to those assays. And this whole process took about 4 months. So I just thought I'd put some of the sort of comparisons between the dye-based DSF and the cell-based thermal shift assays here. So I think the major one is the fact that it's the reagent. So the dye-based DSF, it's a lot of protein that you need. That makes it a no-go for a lot of projects, and that's where the cell-based thermal shift assays come into their own, because they open up the possibility to intractable targets that we can't get protein for. Having said that, the challenges with the cell-based assays is that you require a high-bit tag cell line or antibodies, and both of those are challenging. Making these high-bit tag cell lines takes time, so it's a consideration as well. In terms of equipment, it's nothing special. It's qPCR machine for the DSF and a thermocycler for the cell-based assays and the luminescence plate reader. In terms of scalability, both of these assays are quite expensive to run. So in HTS, we like to screen at 2 to 3 pence per well. And these are, DSF is slightly more than that, and the Setzer is a lot more than that. Because of this, and for the DSF, because of the protein, we limit this screen to a subset of the collection. So although we have 1.6 million compounds we could screen. In DSF, we only screen 100, and in SEPSA, we screen about 500,000. Despite this, it still takes quite a long time to deliver the screen. So as an example, for a more simple biochemical cell-based assay where you're doing, for example, a read, you might be able to run that in 1536, 200 plates a day. So you'd be able to do that in about 10 days. So we're sitting in an expensive assay format with long lead times. But because it opens the possibility of prosecuting intractable targets, that's why we're doing it. One of the limitations we've got here is that we have to run these in 384 well format. We are avidly looking for a 1536 well plate that can be evenly heated, and in the case of DSF, red, but we're yet to find one. So if you know one, let me know. And then the other major difference, of course, is the environment. So DSF, you've got a much more simple model of protein, you're less susceptible to artifacts, and you don't have any of the, you don't need to overcome the challenge of getting into the cell. Having said that, when you're in a cell-based format, you have hopefully got all of your native binding partners present, so you're more likely to mine a biological MOA, potentially. But this comes with its own deconvolution challenges, so you have to have, as I said, a rigorous cascade setup. So that's all we've got. This is Hit Discovery team at AstraZeneca. We have a lot of fun. So yeah, thanks to all of those people, and there's a lot of people that have been involved with these thermal shift assays over the years in high throughput screening and assay development. So thank you to all of those, and yeah, we'd be happy to take any questions.